Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we already discussed the overview of the bacterial cell wall. Now this is the second part of the bacterial cell wall regarded with the cell. And in this second part of the bacterial cell wall, what we are going to talk about? We are going to talk about peptidoglycane. So let me write. Peptidoglycane. We also call that murine. We also call that what? Murine. So this is actually the cell wall. And if you see, this peptido means amino acid. Amino acid. And this glycane means glucose or sugar molecules. Sugar molecules. So actually a peptidoglycane is nothing but it is a two sugar molecules which are connected with each other and make a layer which we call that peptidoglycane, or similarly we also call that cell wall. Fine. So what actually happened, let me tell you a little bit about that. This is, let's suppose, a layer of the cell membrane, this black one, and after that what we have a molecules like that, as I drawing. These molecule means these red and blue one are actually represents what? Peptidoglycane. And similarly we have also just like that. Now these are what? These are the peptidoglycane layer. So peptidoglycane is actually, it is made up of two main molecules or two sugar molecules. We call that NAM and NAG. So how this NAM NAG will be look like? Let's suppose this blue one represents the NAM and this red one represents the NAG. When NAM NAG are combined together, and when another NAM NAG and then another NAM NAG and then the first layer of NAM NAG, second layer of the NAM NAG, when all NAM NAG complex are combined together, then they make what? They make the cell wall, or we can say peptidoglycane layer. Actually, peptidoglycane, if we see the home, means the home wall is made up of bricks. When bricks are combined together, then they make a wall of the home. Similarly, when these NAM NAG are just like a brick, when these NAM NAG are connected to each other and when they are combined to each other, then they make the wall of the bacterial cell. Okay, so they can actually responsible to make the peptidoglycane layer, or they can make the cell wall. So this is about the NAM NAG, and these are the building blocks, or these are the main important material of the cell wall. How it will be look like if we see? So this NAM NAG will be look like in this form. So this is what? This is NAM. There will be a nitrogen, a hydrogen. There will be another hydrogen, and then after that we have what? We have the NAG as well. Okay, so this if you see, the name is actually stand for what? Name is actually stand for N-acetyl, sorry, N-acetyl name, N-acetyl muramic acid. Muramic acid. And this NAG is stand for what? This NAG is stand for N-acetyl glucosamine. Glucosamine. samine. So these are the stand of NAG and NAM. What actually happened with NAM? We have 5 amino acid molecules are connected with each other. So let me draw these 5 molecules, but before that they consist of a carboxylic group. So this is the carboxylic group. These are the carboxylic group. With this carboxylic group, we have another molecules, we call that what? D-lactic acid. D-lactic acid. With D-lactic acid, we have another molecules, L-alanine. L-alanine. In short form, we can write L-ala, means L-alanine. And after that, we have another amino acid, D-glutamic acid. And after that, we have another molecule, we have diamino acid. What we have? We have diamino acid. This diamino acid will be in 2 form. I will explain it further. Then we have diamino acid, and after that we have D-alanine. We have what? D-alanine. So these are amino acid: D-lactic acid, L-alanine, D-glutamic acid, diamino acid, D-alanine. Okay, but this diamino acid, what is the diamino acid? If we see, this diamino acid means 2 amino acid. What does it mean? This means that it contains 2 amino acid. 2 amino. The first we call that lysine. Okay, and the second we call that meso diamino pymetic acid. So these are the 2 amino acid. That's why we call that diamino acid. This lysines will be present in case of gram positive bacteria, and this mesodiamino pymetic acid will be present in case of the gram negative bacteria. Okay, this is present in gram positive bacteria, and this amino acid will be present in the gram negative bacteria. Inside the bacterial cell, we have this cell membrane, and then we have the previous NAM NAG. What actually happened? This peptidoglycane layer will be synthesized when the previous NAM NAG are present. As you can see, this previous NAM NAG is present. That's why the next NAM NAG will be synthesized. If the previous NAM NAG are not present, then the synthesis of the new NAM NAG will be not occur. Okay, so there will be always NAM NAG are present. Previous NAM NAG are present in the bacterial cell wall. Okay, and after that, then new NAM NAG are synthesized. So how the lysines will be look like? The lysine, if you see, the lysines will be look like in this form. What we have the so the lysine will be look like in this form. So this is the lysine. So lysines will be look like in this form, and if we see the structure of this mesodiamino pymetic acid, so it will be look like in this form. Because why we call it mesodiamino pymetic acid? Because it contains 2 molecules of the amino pymetic acid. This means this layer. As you can see, this is actually amino pymetic acid. So it contains 2 aminopymatic acid. You can see aminopymatic acid are 2. So that's why we call that mesodiaminopymatic acid. This will be present in gram negative bacteria, but in gram positive bacteria we have lysine. What actually happen, how actually the means the peptidoglycane are synthesized. This is actually the structure of the peptidoglycane. What actually happen, how the peptidoglycane are actually synthesized. The peptidoglycane will be synthesized. First of all, we have this kind of structure, we call that first of all, let's suppose this is the gram positive in case. Now in gram positive, how the cell wall will be synthesized. First of all, we have molecules, we call that NAM. What we call that? We call that NAM. This green one represents the NAM. And with this NAM, we have a bond of oxygen. And with this bond, we have another molecules, as I draw with the red marker. These are what? These are the NAG. So this blue one, green one represents the NAM, and this red one represents the NAG. Okay, with this NAM, not with NAG. Okay, with NAM. With NAM, what we have? We have 5 amino acid. Two, 3, 4, 5. Yes, and then we have another name NAG. Let me draw here. And they have also the same bond. Now with this name again we have the same 5 amino acids, as I draw with the black marker. 1, 2, 3, 4, 5. Yes. What actually happened, how the cell wall is actually synthesized. First of all, these last 2 amino acids will be chopped away. These last 2 amino acids are actually D-alanine, and they will be chopped away, means cleaved out. And this will be cleaved out by an enzyme, means this peptidoglycane will be synthesized with the presence of enzyme. So we simply call that penicillin binding protein. We call this enzyme penicillin binding protein. How it will be look like? It will be look like in this form. So this is what, this is penicillin binding protein. They have 2 activity. The one activity we call that transpiptidase activity, and the other activity we call that D-allocarboxypeptidase activity. So how many activity? 2 activity. Transpiptidase activity, and the other we called it D-allocarboxypeptidase. So they have 2 activity. First of all, what actually happened, we have a NAM-NAG complex connected with each other by this oxygen molecules. When they are connected with each other, then they make NAM-NAG complex. After that, we have 5 amino acids are connected with each other. Let me write the name of these 5 amino acids. The first one amino acid are called what? L-alanine. This one is called D-glucosamine, and this one is called lysine, and this one is called D-alanine, and this last one are also called D-alanine, but it will be chopped away. The same situation are also present with this NAM-NAG complex. So these 5 amino acids will be connected with NAM, not with NAG. Once these 5 amino acids are connected, then the enzymes, then what actually happens, this NAM-NAG complex with 5 amino acid, with 5 amino acid will be shifted to the periplasmic space, and once they reach to a periplasmic space, then they will be connected with the previous one. These are the previous one, and let's suppose These new represents what? These new one, as I draw with green marker, these represents the new amino means new name neg. This is the new name neg, and this is the previous one. So the new name neg will be attached with the previous name neg. Once these are attached, then another name neg are also bind, connected with each other. We have another name neg complex are connected with each other, and they have also the 5 amino acid, means L-alanine, D-glucosamine, lysine, D-alanine, and again D-alanine. So they are again connected with here. But before connection, what actually we have? We have an enzyme, or we have a protein, we call it penicillin binding protein. This protein comes and they do two kind of activity. The first activity we call the transpiptidase activity. What actually happen in transpiptidase activity? In this activity, they can cleave 2 amino acid. As you can see, this is the one amino acid, this is another amino acid. So they can cleave 2 amino acid from the name. So first from this name and then from this name. So they can cleaved away, means D-alanine are cleaved away. After that, the terminal end, as you can see, they can join this terminal end with each other. And this activity we call that D-L-carboxypeptidase activity. The linkage actually in gram positive, we call this linkage glycine. Why? Because there are 5 molecules of the glycine, means these are the glycine molecules. These glycine molecules are connected and they make a peptide linkage. We simply call that also peptide linkage. We also call that what? We also call that peptide linkage. So once we get the peptide linkage, so then this layer will be strong. Means in this case the layer will be strong. And then another name neg are connected, and then again the penicillin binding protein do their activity, means they again cleave the amino acid and join the terminal portion of this bond, and then we get again a strong name neg, means again we get the name neg, and then another. Finally, what we get a thick layer of the peptidoglycane or a thin layer of a peptidoglycane. So that's how the peptidoglycane, or we can say cell wall, are synthesized. But this is actually in case of gram-positive bacteria. In gram-negative bacteria, what we have, let me remove this one portion. And what actually happen in gram-negative case? In gram-negative, we have also the same name neg are present. Let's suppose in this case, this black one represent name, and this blue one represent the neg, and they will also connected with what? With oxygen molecules. Okay, and with this name, what we have again 5 amino acid. 1, 2, 3, 4, and 5. And again we have the same things, name and then neg. Okay, so this name again contain the 5 amino acid, but but in this case what actually happen, the amino acid are different, although they are the same, means the process of peptidoglycane synthesis are the same as in the gram-positive bacteria. Gram-negative bacteria have the same synthesis of the peptidoglycane. But one thing you should have to keep in mind, the peptidoglycane the amino acid sequencing or the amino acid are actually changed. In this case, what we have the let's suppose this one is called L. What we call it? We call it L-alanine, and this one is called, we call it D-glutamic acid. Let's suppose this is another amino acid, and this third one we call that, this is the first, this is the second, this is the third. The third we call that D means diamino palmitic acid, as I discussed in the previous, diaminopalmitic acid. In this case we have L-lysine, but in this case we have diaminopalmitic acid, means mesodiaminopalmitic acid. And after that we have again D-alanine, and this is again D-alanine. So two molecules. Okay, so this is the combination. But what actually happen, how this linkage will occur in the gram-negative bacteria? In gram-negative bacteria, the linkage will be occur in case of, means this is 1, this is 2, and this is the third, and this is the fourth. So the fourth one will be connected with this third one. Okay, so this fourth one means this fourth one amino acid will be connected with this third one, means the mesodiaminopalmitic acid. So they will be connected with the mesodiaminopalmitic acid. So this D-alanine, the terminal end of this D-alanine and the terminal end of this diaminopalmitic acid will be connected with each other and make a strong linkage. Okay, so that's how they can synthesize the bacterial cell wall, or we can say peptidoglycane layer. I hope you understand, but if still you have any kind of confusion about that, so write it into a comment. Inshallah, we'll give you answer as soon as possible. I already explained this peptidoglycane or synthesis in beta-lactam antibiotic. If you missed this video, if you not understand, although it is very complicated, but if you not understand this video lecture, so go back to my channel and just search beta-lactam antibiotic. And in this video lecture, in that video lecture, you will easily find the that how actually the peptidoglycane are synthesized. On that video lecture, actually I explain it very well and I simplify it. So I explain in an easy way in that video lecture, but in this video lecture it is very complicated. So still I hope so still you can also get the idea from this video lecture as well. Okay, so that's all about the peptidoglycane and everything related with the cell wall and how actually the cell wall are synthesized. I hope this videos help you. Inshallah, in the next video lecture we are going to talk about gram positive cell wall and gram negative cell wall. Okay, so please stay tuned and continue to watch the rest of the video regarded with the bacterial cell wall. Thank you so much.